Hi everyone, I hope all of you are doing great. So today we will discuss about flow cytometry, which is a very useful tool, especially who are working with biological sample. So what is flow cytometry? It is a system for detecting and analyzing the cells or particles in liquid stream when a laser beam pass through that liquid stream or like the cells or particles of that liquid stream and then illuminate beam. So it has basically four parts. One is fluidics, another one is optics, then detection and analyzing part. In this video we will discuss about the fluidic system of flow cytometry, and we will also the hydrodynamic focusing is included in the fluidic system. So we will also discuss about the hydrodynamic focusing, which is the essential part of flow cytometry. In flow cytometer, cells or particles to be analyzed must be suspended in liquid that flow through the cytometer. You can see here that it is suspended in fluid and it is flowing through this way. So it is mandatory to suspend any fluid. The selection of liquid depends on the nature of particle, such as the liquid can be like saline, water, milk, or even beer. So it actually depends on the particle nature. There is some upside and downside of this system, of this fluidic system. The upside of fluidic system is it can analyze a lot of cell in a short period, and another upside is it can give the statistical information about large cell population, again in a short period of time. But there is downside. The downside is particle must be single. If it is blood, bacteria, or small plankton, it's okay, that's not a matter. But for the tissues, the tissue always stay in aggregated form. So when anyone need to do flow cytometry test for tissues, the tissue should be disaggregate, and when they disaggregate, the tissue architecture related information can be lost. There are two methods to deliver cells into laser beam. One is, if cell suspension is in a test tube, we can give air pressure to the test tube to force the cell out. And another method is, we can take the cell suspension into a syringe and then we can give pressure by using the syringe plunge. The purpose of both of this method is to getting optimal optical sensitivity. And for getting optimal optical sensitivity, the flow characteristics should be, there should be two flow characteristics. First flow characteristics should be, when cell get to the laser beam analysis point, they should be look like beads spaced out along a string. That means at the center of the laser beam, there should be a specific distance in between two cells, and the distance should be the similar distance in between all cells, so that they can illuminate brightly and uniformly. The second characteristic is the flow rate of cell should be slow enough so that all cells can get enough time to get in, so that they can produce intense signal. The illumination is very important to produce detectable signal. So if cells get enough time to get illuminated, then they can produce detectable signal. Now we will talk about the hydrodynamic focusing, and by this way cell confining occur. So when hydrodynamic focusing happen, actually here you can see that this is the sheath fluid and this is the core fluid. So when the volume of sheath fluid is higher than the volume of core fluid, then hydrodynamic focusing happens. This hydrodynamic focusing actually create a fluid system where cell move forward in a very discrete pace. Cell always stay in the middle position. The faster moving sheath fluid, it actually force the hydrodynamic core so that particles move in the same axis at same velocity. So the key information is that because of the hydrodynamic focusing, cells are stay in the middle position in a specific distance in between them at a specific rate, so that at a time only one cell or one particle pass the laser beam, and it reduces the rate of coincidence so that we can get the data without any overlapping, and eventually it is a good data. So the hydrodynamic focusing is very important and playing a very important role here. But there are some factor which can impact, which can affect the hydrodynamic focusing or the flow rate of cell. Here you can see that when pressure is low, core stream is narrow, and only a single cell or single particle is passing the beam, the laser beam, at a time. But when the differential pressure is higher, at that time, because of the high pressure, a lot of cells is passing through the laser beam at a time, and eventually the core stream diameter is also wider. So this means that when pressure increases, when we inject the sample into the core stream at a higher velocity and the pressure is higher, it causes the wider core stream diameter, and because of this, more cell passes the laser beam at a time, and it causes coincidence and signal overlapping or peak overlapping, and ultimately which result we will get, the quality of the result will not good. So here you can see that in traditional hydrodynamic focusing, where flow rate is higher and also the pressure is higher, this is the problem, that a lot of cells are passing the laser beam at a time. This problem can be overcome by using the acoustic field. When we will use the acoustic field, then acoustic field assisted hydrodynamic focusing. In acoustic field assisted hydrodynamic focusing, sound wave forces the cell to remain aligned and pass the laser beam one at a time. Again, if the sheath fluid velocity is higher, it is very advantageous for the flow cytometry because it helps in faster cell analysis. Again, when the sheath velocity is higher, it helps to maintain narrow core diameter, or ultimately it helps to avoid coincidence. So from this discussion, we understood one thing, that is that in flow cytometry, if we want to get a good result, then cell flow rate or particle, whatever you are analyzing, the flow rate is very important. If we can maintain the flow rate in a specific manner, then we easily will get a good result without any coincidence or any overlapping. There are three factors that basically can affect the cell flow rate. One is sheath stream velocity, which we discussed already. Another one is core diameter. We also discussed about it. And the third factor is the cell concentration in the original sample suspension. These three things actually affect the flow rate in the flow cytometry. Another point is that flow chamber design is very important in flow cytometry. the flow chamber is used there, and the design of flow chamber is very important. There are basically different types of flow chamber is using, such as cuvette style flow chamber and jet in air chamber, and there are also some others, but those two types are using mostly. There are some importance of this of the design of this flow chamber, because this flow chamber helps to reduce the turbulence, helps to reduce the light of background scatter. It also helps to maintain the stream velocity, and also helps to maintain the narrow stream in the core diameter. However, so in this video I wanted to give you some very vital information about the flow cytometry, and also the very important some information about the very important part of flow cytometry, which is fluidics. And I also try to describe you the hydrodynamic focusing, and actually this is the basic information about the hydrodynamic focusing what I discussed. So I hope it will help you to understand the flow cytometry and its fluidic system very easily. So see you in the next part 2, where I will discuss about the optics, and in the future in other parts. So till that time, stay happy. See you again. Bye.